Hi, everyone, and thank you for joining me today for episode 692 of the People Behind the Science podcast. I'm your host, Dr. Marie McNeely, and today I'm looking forward to revisiting our conversation with our guest, Dr. Mike Blatt, originally published on February 20th of 2017. Listeners, Mike is a cell biologist and physiologist who has been recognized internationally for his research on the physiology of plant cells, including their ion channels, vesicle trafficking, and mechanisms of gas exchange. He is also passionate about electronics, and he's actually built a lot of the equipment that they use in their lab for research. And in this episode, listeners, you're going to hear more about Mike's life and his research, so get ready to meet another one of our wonderful people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, our guest is Dr. Mike Blatt. So, Mike, how are you today? Welcome to the show.
Fine. Thank you very much, Marie.
Excellent. Well, we are excited to chat with you today, and we're excited to learn more about your life and your work. But I want to start by giving our listeners just a little bit of background information here before we dive in. So, listeners, Mike is the Regis Professor of Botany at the University of Glasgow and Adjunct Professor at Pennsylvania State University. He conducted his undergraduate studies at Simon Fraser University in Vancouver and at the University of Wisconsin-Madison, where he received his bachelor's with honors in botany and biochemistry. Next, Mike was awarded a PhD in plant biology from Stanford University while working in the Department of Plant Biology at the Carnegie Institute of Washington. Now, during his graduate work, Mike received a Fulbright Hays Graduate Fellowship to study at the University of Nuremberg, and afterwards he traveled to Yale University Medical School to accept an NRSA postdoctoral fellowship, and then on to the University of Cambridge to accept a NATO postdoctoral fellowship. And Mike has served on the faculty at the University of London and Imperial College London prior to joining the faculty at the University of Glasgow, where he is today. And he is the recipient of many awards and honors over the course of his career, including being named a Fellow of the Royal Society of Biology, the John Simon Guggenheim Memorial Foundation, the James Hutton Institute, and the Royal Society of Edinburgh. And Mike is also editor-in-chief of the premier international journal, Plant Physiology. And listeners, Mike is here with us today to talk about his life and talk about his research. And we want to get to know you, Mike, today, both as a scientist, but also as a person. So can you start by telling us what you like to do when you're not doing science?
Well, I have a lot of interest, but I have to admit one of the things I love the most is winter sports.
Excellent.
Especially downhill and cross-country skiing, something I've been doing for pretty much all of my life. Lately, my wife and I have started ski touring, in fact, but we're off to the Alps in the next couple of weeks, and I'm taking my father with me. He and I have skied throughout much of my life, and believe it or not, he still skis. He's 92.
Oh, wow. That's amazing. So do you aspire to continue skiing until you're in your 90s?
I aspire to, shall we say.
Yeah. Well, fantastic to hear a little bit more about what occupies your time outside of science. Now, I know there's probably not a ton of skiing near Glasgow. What's your favorite place to go?
Well, we do have skiing here. It does snow, but you have to be pretty fast to catch the snow. Otherwise, it's rock skiing.
Yeah. Could be fun. New sport.
Yeah, absolutely.
Well, Mike, now tell us a little bit more about your scientific side next. What is this high-level description that you give people when they ask that question? What do you do?
Well, I mean, it depends upon who's asking the question, but more often than not, it's individuals who don't have much of a scientific background and often actually don't associate research with teaching. So usually I start off by simply saying that I teach at the university. And actually, I enjoy teaching very much, especially engaging with the students early on. As much as anything else, it's a matter of entertainment, especially at the first 2 years as undergraduates. But you really know when you capture their imagination. You see it in their eyes and the way they challenge you. And I always find that a real thrill. Otherwise, apart from lectures, tutorials, supervising students in the lab, these things keep me very busy. And of course, being editor-in-chief of Plant Physiology also keeps me very busy. But when I'm not engaged in teaching, administration, etc., I'm in the lab. I really enjoy being in the lab myself. I think it was C.P. Snow who once said that science cannot be delegated. And I have to admit, I agree with his statement, although I think for different reasons. Mm-hmm. So if I'm explaining the research, I would say that I just enjoy taking things apart and putting them back together to see how they fit together. And this applies to cells as well as laboratory equipment, probably much to the dismay of my colleagues.
Certainly.
But one of my real pleasures is electronics. So a lot of the equipment in the lab I've built up myself over the years, particularly for the electrophysiology.
Very cool.
As far as the research is concerned, I would classify myself as a cell biologist or a physiologist. And I often refer to mechanical analogies to explain. So I would say that if I were a Martian bicycle cell biologist or physiologist, I would be studying how the bicycle is assembled, how it works, and most importantly, how the parts fit together to actually do the work that a bicycle does. And I think it's the last bit that I find most exciting, because it's in the way things interact that you discover the emergent behaviors that are not obvious in the parts when you look at them. And these emergent behaviors really only appear when you start putting the parts back together. And these are basically what we associate generally with life.
Certainly, I think in many cases, the whole is so much bigger than the sum of all the parts going together.
Yes, definitely.
Well, great to hear more about your work. I really like here that you brought up all of the different roles that you have as a faculty member, because it's not just the research. You're balancing all of these different sides of the job that you do. And I think it can be a challenge sometimes. And I love using quotes to motivate me and keep me going. So do you have a favorite quote of your own?
I always struggle on this one. It's easier to point out people who provide good quotes. I think my favorite is Oscar Wilde when it comes to that. I think he made an appropriate quote as far as quotations. He said, "Quotations are a serviceable substitute for wit."
Ooh.
And I find that is very useful at times. But he has quite a few others. So experience is the name that everyone gives to mistakes. And the pure and simple truth is rarely pure, and it's definitely never simple. And of course, my favorite, I suppose, is the mystery of the world is in the visible, not in the invisible. So I think that's about the best I can do for you.
Well, you provided many different motivational quotes for us today, listeners. Pick your favorite. And I love talking about the different things that motivate scientists. And I think along the course of your career, it's not just the experiences that motivate you. It's not just the projects that you're working on. Oftentimes, it's the people who have a really big impact on you. So when you think back, Mike, are there any people that really jump out in your mind as people who inspired you to go into science or served as mentors or role models?
Yes, there are certainly some real role models that I've had over the years. I was really very fortunate to have, as a PhD supervisor, Winslow Briggs, and also unofficially Wolfgang Haupt, who supervised me when I was on the Fulbright Hays Fellowship in what was then the Federal Republic of Germany. They're very different people, of course, but in my experience, both men sought to help everyone in their care to find the very best in themselves, and both imbued these efforts with a lot of humor. I recall Winslow, I don't think he'll mind if I mention this, on one occasion setting to rest my own concerns about my negligence by recalling one experience that he'd had when he was a PhD student, and he'd been responsible for the amphibian rooms, but forgot to replace the tops on the tanks.
Oh, dear.
Yes, quite. As Winslow put it, the next time he went in the room jumped.
Oh, jeez.
You can imagine the task of trying to recapture several hundred rather agitated toads.
That is a challenge, absolutely.
So Winslow and I love to play word games, and this always provided me with a great deal of amusement. I think it had provided Winslow with a lot of amusement too. So I managed to slip a few malapropisms in my PhD thesis, though I suspect he probably turned a blind eye.
Right.
However, I was very pleased to see that one of them came back to me in an acknowledgement of one of his recent publications that I edited for Plant Physiology.
Oh, that is fantastic. And I don't know if you know, we've actually had Winslow on the show not too long ago. So he has told some excellent stories.
I also had, just to mention one other person, and that is Enid McRobbie. She was my supervisor when I was on the fellowship and then as a senior postdoc in Cambridge in England. She is just an amazing person. I mean, one of her great strengths, also had a great sense of humor, but one of her great strengths was her seemingly effortless ability to guide students and staff in the lab without even directing them. She had her own research projects, and she would periodically be at the bench herself. But she let me and everyone else in the lab work on our own and gain from our own experience through trial and error. It was just a remarkable guidance that she provided. The other thing that really stands out about Enid is that she operated on the premise that she must have contributed directly to generating the data for a paper before she would consider being included as an author, for whatever motives, but it really gave those in her lab an added sense of ownership and identity with their work. And I think it takes real inner strength and principles to approach laboratory management in that way.
Certainly. Well, wonderful to hear about these people who had an impact on you during your scientific career. But I'm curious, Mike, to hear about where it all began. Can you think of the point in your life where you started to get interested in science?
I think I've been interested in science right from the very start. For whatever reason, even also with plants, even as a pre-adolescent. My father was a physics professor at Midwestern University, so much of my early childhood memories with him are helping, inverted quotes, of course, in his lab. And I suspect that's where I developed a real delight in tinkering and all sorts of things mechanical. But my interests were always in biology. If I think back, I suppose my interest in plants, or the roots of them at least, if you'll pardon the pun, took hold in the excursions that we often took hiking in the Rockies over several summers and skiing in the winters. I'm a keen photographer too, and that was also true even before my adolescence. I had an old Pentax SLR camera back then, and I'd take these on the trips. I think I probably just had a different eye from what you might expect of an average teenager.
So are you still an avid photographer today?
I am still an avid photographer today. My wife sometimes despairs when I take over the kitchen and turn it into a dark room, but there you go.
Right. Make your dinner next to your little pans there.
When all the food and everything else goes out, I'm working with the chemicals.
Good. You've got the basic laboratory procedure there. Well, wonderful to hear about these early days of your career. But can you tell us, Mike, were there any big turning points or key moments along the way that really helped you get to where you are today?
Well, I suppose when I left Simon Fraser, I was very focused on biochemistry as an undergraduate at Simon Fraser, but when I left Simon Fraser and moved to Madison, I found a second home with some of the folks at Burge Hall, what was then the botany department. And I had some pretty amazing teachers there too. Folker Skoo was there, Ray Everett, Eldon Newcomb, and they really solidified my interest in plant form and function. And I think that was, in a sense, the start of my interest in plant biology research. So although I went to Madison thinking I would do a degree in biochemistry, I came out, as you noted before, with 2 bachelor's degrees, one in biochemistry and one in botany.
Certainly. And I know you traveled quite a bit throughout the next stages of your training. So can you tell us a little bit about those steps along the way?
Well, I went straight into the PhD studies at Stanford after Wisconsin, and at the time we rotated, and I had the opportunity to rotate to the Hopkins and Marines labs, which were down the coast near Monterey, and also to the Carnegie Institution of Washington, which was right next door. And I have to admit, it was very tempting to go down the coast, scuba diving and such. And in fact, I did quite a bit of that later on. But because of my interest in plant biology, I stayed on the Stanford campus. So in fact, I didn't really do a lot of traveling until my 3rd year when I went off on the Fulbright Hays Fellowship. But I have to say that the Stanford period was also an interesting one to begin with, because I started with a 6-month rotation with Peter Ray, who at the time was working on isolation and fractionation of plant cells. And with Peter, I worked out a strategy to isolate pure plasma membrane from plant protoplasts. We were aiming for a method that would give us bona fide, absolutely pure plasma membrane, and I don't think it was particularly successful. And most of all, it was far too labor-intensive. The method was to make use of lectins. And I recall starting experiments each week at 4 a.m. on a Monday morning.
Oh, my goodness.
In what ended up being a really brutal 20-hour preparation and extraction procedure. But I did learn an awful lot from that, and it was enough to turn me to photobiology.
Right. Perhaps learned you didn't need as much sleep as you thought.
No, absolutely. We were, I think, on the right track, focusing on the glycosyl residues and the surface of the plasma membrane, although we weren't particularly successful. Larson's group came up with a very similar method, although much gentler about a decade later, and his techniques are tremendously successful. We still use them in my lab.
Gotcha. Well, fantastic to hear about some of the earlier experiences you've had in your career. And I know now you are there at the University of Glasgow doing amazing work. So can you tell us about one of these projects that's ongoing in your lab right now that you are just the most excited about?
Well, I have been working on a project relating to membrane traffic. This is really a project that started more or less, how shall we put it, purely by chance more than anything else, serendipitously. I had a PhD student when I was at Y College many years before. This is going back to the mid-1990s. And at the time, I was very interested in spreading the focus of my lab away from electrophysiology, which is what I had focused on for the better part of 15 years, into something that was a little bit more molecular. And I have to admit, I was not a particularly good student in molecular biology. But in 1995, Barbara Lehman joined my lab, and she had come from the VIB in Ghent in Belgium with quite a bit of molecular biology knowledge. So we put our heads together, and she agreed to take on the molecular biology side if I would teach her electrophysiology, and vice versa. And as I said, I was a pretty poor student. But out of this, we aimed to pull out an ABA receptor using an expression cloning strategy. And in the end, what she pulled out was a protein that looked a little bit like proteins that had been identified as what are called snares. Now, at the time, snares were completely unknown in plants. In fact, it had only been about 3 years since Sunoff and Rothman had proposed the snare hypothesis that these proteins were part of a large molecular weight complex that would assemble and drive membrane fusion. And we pulled it out using an expression cloning strategy that essentially would identify proteins that regulated ion channels. So we were really stunned that there could be some connection between ion channel regulation and what we assumed it might be a snare protein. And that really has lifted the focus of my lab away from guard cells and electrophysiology to a certain extent to membrane vesicle trafficking. So one of the really exciting developments over the past few years came when we identified the binding site on the K channels that interact with these snares. Now, just to put you in picture, the K channels that we work with are assembled out of tetramers, so 4 homologous polypeptides, each polypeptide comprising 6 transmembrane alpha helices. And of those, the last 2 line the water-filled pore through the channel, but the first 4 act as a sort of a semi-autonomous voltage sensor domain.
Interesting.
Yeah, well, now the mechanics is rather interesting. So the KV channels, which are these channels that interact with the snares, their gating has been worked out very well. And we know that now from much studies, both in animals and plants, etc., that the VSDs move in response to membrane voltage. So within the VSD, one of the helices contains a regular series of charged residues, and these charged residues effectively sense the voltage across the membrane and drive the movement of the VSD assembly, these 4 transmembrane helices, partway across the membrane. And then one of those helices is connected through a linker to the last 2 helices, which line the pore. So it's a little bit like these toys that children play with called Hampelmann.
I'm not familiar.
Oh, okay. It's a wooden toy and it has a little string underneath. You pull the string, the strings are attached to the legs and the arms legs and arms swing out.
Oh, yeah. Okay. I never knew the name for that. That's awesome.
So there you go. It's a very bad analogy, but it's essentially the way that the ion channels work. The VSDs pull on the pore and they pull the pore open in order to allow the ions to pass. So the eureka moment for me came, this was about 5 years ago, when a very talented postdoc of mine at the time, Chris Greffen, started pulling out the sequence for the binding site for the channel that would interact with the snare using the technology that we developed of split ubiquitin screening using a rather novel modification of it. And what he discovered was the binding site was at the cytosolic base of the first alpha helix of this voltage sensor domain. So it was right on the cytosolic surface of the VSD. And we both realized right then and there that it was very likely that that binding could be modified by voltage because of the location on the protein. In other words, what we were postulating at the time was that the snare commandeered the voltage sensor of the channel in order to sense voltage and adjust vesicle traffic accordingly. I don't want to bore you with all the details in terms of how we went about proving that. Needless to say, it was a number of delightfully subtle approaches that we resorted to prove the point, and the paper appeared just barely 15 months ago. It's, if you want the reference, it's Greffen et al. 2015, Nature Plants, Volume 1, 15108. And there's also a really nice commentary to go with it from Rick Hoosman and Tom Bisseling. Mm-hmm. But just to put this all in perspective, you might ask, well, why is coupling to a VSD so important? The thing is that plant cells use highly electrogenic proton ATPases to energize the plasma membrane in order to drive solid accumulation, potassium accumulation, and such for cell expansion. And one of the big questions that has been puzzling plant biologists for over a century is how do plants regulate the processes of solute uptake during cell expansion? How do you control, or at least coordinate, uptake with the increase in cell surface area, which has to be provided by membrane fusion through, for example, snare proteins, and also how do you remodel the cell wall, again requiring vesicle traffic and secretion. And here suddenly we had essentially handed it to us on a plate through this interaction, a mechanism by which these two processes could regulate one another. We already knew from Chris's earlier studies that the channels were regulated by interaction with the snare. The snare binding would modify the way that the channel behaved. And here we now had evidence that voltage sensor domain of the channel could also be commandeered effectively by the snare protein to set the poise for vesicle trafficking to coordinate the two processes. It's a really elegant mechanism and shows just how plants have evolved to bring two very different processes together in a rather unexpected way for mutual control. And I have to say, being at Glasgow, I like to refer to this as the governor hypothesis. I'm sure you've heard of James Watt and his steam engines. Mm-hmm. Well, the mechanical governor which he used to drive his, or at least to control his steam engines, it was not something that he invented. It was written up by Maxwell about 30 years earlier. All he did was to realize he could use a mechanical governor as a means to regulate his engines, which I have to say otherwise had a tendency to explode.
That's a problem. Yeah. Minor detail. Well, Mike, fantastic to hear more about your work. And I think the research you're doing, it's so important for many different aspects of function in plants. This membrane trafficking is critical for getting nutrients, for the use and management of water in plants. And these things are so important for agriculture and all of these big problems that we're facing in the future with managing food supply.
No, absolutely. I think the whole issue of water control brings us back to the guard cells. And there again, as I said at the beginning, for me, one of the most exciting things is with all the information that we now have about how guard cells work and how they function, and the sorts of information that we're getting now at a molecular level, we can start to piece these together and look at how those parts fit together to make the whole.
Certainly. Well, great to hear more about your work. And I think we've talked about a lot of the exciting elements of your career so far, but we haven't chatted too much about the challenges and the struggles. And I know, Mike, every scientist goes through them. So I want you to tell us a story next about one of these times where you were struggling with something and how you worked through this challenge.
Well, on the whole, in terms of in the lab, I think it's always been a question of identifying when to give up a project and when not to give up a project. But occasionally you're faced with challenges which you just have no control over whatsoever. And there's one in particular that I can think of. I wouldn't necessarily classify it as a failure, but it certainly was a disaster. 4 months after I moved up from Y College, the University of London, up to Glasgow in the autumn of 2001, the power building, which houses the plant science group here, burnt down.
Really?
Yes, and it was just after the work to refurbish the extension that my lab was occupying had been completed, and we had most of the equipment in place. I mean, imagine, just getting started.
Oh, no. So what did you do?
Well, I mean, overnight, the entire plant science group of 8 PIs and about 50 postdocs, PhD students, and support staff were basically homeless with years of research that basically went up in smoke. Actually, I should modify that ever so slightly because it didn't go up in smoke. It was drowned.
Oh, because of the sprinkler systems.
No sprinkler system. We were standing outside the building for most of the afternoon watching the firemen deal with it. And at one point, one of my PhD students at the time came up to me and he said, there's an awful lot of water being poured into the building, but there's not much coming out.
Oh, no.
I learned an awful lot about fires and the devastation they can wreak over the next few weeks. My goodness. The most common cause of damage in a fire is not the fire itself, but the water used to douse the fire.
So was anything salvageable, or did you have to start completely from scratch?
Well, I was in the building over the following days. We were even up on the second floor wading through 20 centimeters of water, surveying the damage. And finally, when we managed to extract the larger pieces of equipment, which was some weeks later, We even found mold growing on the lenses in the spectrophotometers.
Oh, dear.
Nothing worth salvaging. But oddly enough, I didn't actually, after the initial shock of, oh my goodness, the building's burning down, I don't think I really panicked. It was just sort of a sense of the need to get on with the job. In general, I find if I keep myself occupied physically, there isn't room for panic.
I agree. I'm the same way. If I keep myself busy, there's no time to panic.
Luckily, Glasgow had very good buildings insurance, which covered material losses, the building, and also income, which included postdoctoral and PhD student funding at the time. So in retrospect, if it hadn't been for that support, I think my career as a researcher and that of everyone else in the plant science group would have come to an end there and then.
Wow. Well, Mike, thank you so much for sharing the story. I think you're absolutely right. Sometimes these things happen that you have absolutely no control over, and you have to just pick up the pieces and go.
Absolutely. But there are a few things I learned from that, Marie. I just might share these. So one of the things that you have to do when you're in a situation like that is sort of think sideways. Inevitably, your research at that point and research output will hit rock bottom. And needless to say, all of our group struggled in that respect. But I realized that one of the things that I could do is I could send my postdocs elsewhere. So I sent one postdoc to another lab in the UK and 2 went off to labs in Germany. And several of my colleagues followed suit. And the other thing I discovered is just how important it is to distribute scientific materials within the research community. I wrote back to a lot of colleagues saying, hey, by the way, remember that construct that I sent you last year? I don't suppose you could send it back to me again.
Right. The importance of email. Yeah.
Absolutely. So I was able to reconstruct a lot of the work that Barbara Lehman in particular had done, and others in my lab this way.
Gotcha. Well, Mike, thank you so much again for sharing the story, but we don't just want to dwell on the difficult times in your career. We'll let you tell a success story next. So can you tell us one of these wonderful moments, whether it was a big one or even just a small victory that meant a lot?
The story with the binding site on the VSD was certainly a big victory. It's one of those moments when you think, wow, I can see the answer straight away. The hard work comes ahead, but you have this image already of how it's almost certainly going to work. And it's just such a delight when you see things fall into place. Sometimes some parts don't, and then you have to rethink. But it's that sort of clarity of vision and realizing what almost certainly is staring you in the face, which is such a big eureka moment. But there have been lots of other ones. One of the things that always amazes me is when a piece of equipment goes down and I'm saying, oh my goodness, what am I going to do now? I can't sort it out. I can't figure out where the fault is. And more often than not, it's going to be some intermittent fault. So the little successes in these cases are just sort of solving those sorts of problems.
Mm-hmm. Well, I think I take the time to celebrate every success, no matter how small. So I think that is important for keeping you going as a scientist.
That's what the tea room and the coffee room is for.
That's right. So do you have a favorite way to celebrate your successes in the laboratory?
We don't have necessarily a tradition for successes per se. What we do is we celebrate when a paper is published with a small cake or something like this during lab meetings. My lab gets together once a week. We cover journals and we also occasionally have presentations and such. And often these are accompanied by 1 or 2 cakes presented by the postdoc or student who has published the paper.
Ooh, they have to bring their own cake.
Oh, absolutely. This is a tradition that I picked up from Enid's lab, actually. We used to go down to the shop around the corner where they sold sticky buns, as we used to call them. And the funniest part was you'd go in there and you'd ask for sticky buns, and the lady behind the counter would look at you with this very dour look on her face, and she said, they're not sticky buns, they're Chelsea buns.
Ah, of course.
So that half the fun was looking at the face of the poor student or postdoc who, for the first time, got a paper published and had just come back from the shop.
Demoralized completely from their experience. I love it. Well, Mike, we've talked a little bit about your laboratory, but also some of the things that you do when you're not in the lab. And I love hearing about what scientists are reading when they're able to escape the scientific literature for a little while. So do you have a book that you can recommend for us that you've really enjoyed, whether it's a science book or not, for me and our listeners today?
Well, my reading is rather eclectic, I have to admit. Of course, as I said, I'm a big fan of Wilde and his plays, but I really like just good writing generally. I had great fun reading Steven Pinker's recent book, The Sense of Style. For me, at least, it's simply laugh out loud fun. And if I think of a writer that I like, one that I've gone back to read time and again, it's Ursula Le Guin. She's classified often as science fiction or fantasy, And I do enjoy that genre very much. But for me, her writing really stands head and shoulders above many other modern fiction writers in that she really captures worlds both outside and also inside the soul with such remarkably sparse prose. Just amazing.
Awesome. So do you have a favorite book from Ursula that you can recommend?
The Telling, beautiful book. The Earthsea, well, I guess it's a quartet now. It's not a trio anymore. Also a remarkable series of books.
Fantastic. Well, I will put these on our website for our listeners to find if they are searching for their next great read. And I know we've talked about some of the different places that you've been, Mike, throughout these different parts of your scientific career. And I think travel is a huge part of the life of a scientist. So if you think back to all the places you've been, do you have a favorite place? And can you tell us what you did there and why it was so awesome?
Gosh, well, I don't know about a favorite place, but I was at a meeting 2 summers ago, Pan-American Plant Biology meeting, held in the most unexpected place you'd expect for a biology meeting. It was in the middle of the Atacama Desert in northern Chile.
Oh, really?
Yeah, it's the driest place on Earth, apparently, and it certainly was very dry. I'm told that the desert does bloom about once every 3 or 4 years after it rains. It only rains once every 3 or 4 years. But otherwise, it's basically a moonscape. And the meeting was in San Pedro, which is in the middle of the desert. You land in an airport not too far from the very large mining operation in northern Chile, and you have to drive a good 150 kilometers to get to San Pedro. It was a trip on an absolutely ramrod straight road. Went up over a pass at 4,500 meters. And all you see, as far as the eye can see to the horizon, and when the air is dry, you can see a long way, it's absolutely nothing but rock and dust.
Wow.
Surprisingly beautiful.
So would you go back, Mike?
Oh, absolutely.
Yeah.
I thoroughly enjoyed that experience.
So were you just there for the conference, or did you get to stay and enjoy it a little?
Just at the conference. I had colleagues in Santiago that I went to visit beforehand. I spent a little time in Santiago afterwards, but then I had to get back to the UK because it was in the middle of the teaching session.
Gotcha. Well, at least you could enjoy the long ride from the airport to the conference center.
Oh, it was amazing.
See the beautiful scenery.
Did a fair amount of trekking around during the conference as well and visiting some of the more interesting sites nearby. And the first day I was there and 3 other people at the conference rented bicycles and we cycled 40 kilometers to one of the local sites, which was an amazing experience in itself. My goodness, my rear end was sore after that, though.
I can imagine. That's quite a long bike ride. Well, Mike, this sounds like an amazing trip and a wonderful place to go that you might not have expected to go as a plant biologist. Absolutely the middle of a desert, perhaps, is not the first place that you would think of where you would be holding a conference, right? Perhaps a rainforest or something. But, well, thanks so much for sharing this story. And I think we've talked a little bit throughout our conversation about some of the people that you've worked with throughout your career. And I think the people are part of what makes science so wonderful. And I think there are stereotypes out there about what scientists are like, but we like to try and break those on our show or show this human side of science that I think doesn't always come through. So I think in different labs you work in, there's quirky traditions, or you work with people who have amazing personalities and senses of humor. So do you have a story, Mike, of this human side of science that you can share with us today?
I don't think we have any quirky traditions, although we have some pretty quirky people. I do love wordplay, and I have to say, Marie, usually I have to work at it quite hard, especially if you want to get something in print surreptitiously.
Right.
So the ones that are really amazing are the ones that happen spontaneously. I was delighted to see when our paper, Nature Plants, appeared that our initial abstract, the first version that came out, referred not to the trafficking, but to the trafficking in plants.
I love it.
Not only a paper in Nature Plants, but royal anointment to boot. I felt obliged to point the editorial error out to the journalists, but I did regret the fact that they changed it 24 hours later.
Right.
I can give you another example, too. My computer guru, Adrian Hills, who works with me, I have to say, is much better at this sort of thing. And when we travel together, which we've done on quite a number of times, I'm usually in stitches half the time. He's the one who developed our electrophysiology software that we use, and also the modeling platform that we developed over the past few years. And I think I mentioned before, but he has a very eclectic naming tradition, and his names are sometimes quite funny. So, for example, the electrophysiology suite in the lab is called Henry.
Ah.
We use the 3rd right now.
Yeah.
He's working on Henry the 4th. I think it's part 1. And I have no idea what he's going to do when he gets to Henry VIII.
Right. Now, is Henry an acronym or just a name?
I asked him at one point why he chose the name Henry, and he looked at me with his absolutely straight face and said, because his uncle is named Fred.
Oh, jeez. Fantastic. I'm definitely a sucker for wordplay and puns myself, so I'm loving all of this, Mike, I must say. And now we've chatted a little bit now about some of these people you work with, and I think it's amazing to have this vibrant community of scientists that you're able to collaborate with these people in your laboratory. And I think it's fun to talk to them about these big questions in science, not only the projects that you're working to answer right now, but the ones that you aspire to answer in the future that you would love to answer if you had everything. So I'm going to give you the chance, Mike, today to tell us, what is this big question you have in your mind? One of these things that if I gave you all the funding, staff, technology, feasibility, whatever you need, what would you want to answer?
Well, it's a big challenge, I must say. I think one of the things I would certainly want to go after, I don't know that there's any one thing that would be sort of stand up, but one thing I certainly would want to go after is the other class of ABA receptor, not the phosphate cascade. The discoveries of the START family that Sean Cutler and others made a decade ago has really given the field a boost in guard cells and water relations, etc. But there's pretty good evidence that there's another class of receptors out there just waiting to be discovered, and we just don't know anything about it yet. So that's one of the things that I would want to go after. I think the other thing that really has excited me in recent years is optobiology. I would very much like to be involved in developing an optobiological tool that would measure membrane current, not just voltage. I think that kind of development is not too far off, and I think it would certainly revolutionize the way that we approach, for example, physiopathologies in plants as well as diseases in humans.
Fantastic. Well, I like both of these questions. I'm going to hone in first on this first one. Now, you mentioned this elusive other class of receptors. How do you even go about trying to find it?
Oh, gosh. Now, well, if I had the answer to that, Marie...
You would have found it already, right?
Exactly. I would have found it already. I think sometimes you just get lucky, and that's all there is to it.
Gotcha. Well, Mike, thanks so much for dreaming big with us today and talking about these big unanswered questions in your field. And I think over the course of your career, you not only get to answer some of these really exciting research questions, but you work with amazing people, as we said. And these people are great sources of advice to help you get through your career. So can you think back to all the people that you've worked with? And is there one piece of advice that someone shared with you that really helped you that you want to pass on to our listeners today?
Well, along with Enid at Cambridge, Tom Ap-Rees, who was at the time head of the department, was a great supporter of mine. And his mantra, although it wasn't really advice for me personally, but his mantra was always make the best out of what you are given. And in retrospect, I think that was certainly very useful for me, both at my time at Y College and since. I think the other piece of advice that I would give anybody, and certainly give to folks in my lab and colleagues, is to be prepared to reinvent oneself at least once every 10 or 15 years. Most of the people that I know who have really been successful throughout their scientific careers are ones who have undergone at least one or maybe two or sometimes three seismic changes in the way they approach science.
So have you reinvented yourself lately, Mike?
Yes, I recently reinvented myself as a modeler. About 5 years ago, we developed the OnGuard software platform that I mentioned before, which is essentially a systems biology platform, but a quantitative one for modeling guard cells. And that platform has really opened up whole vistas on how transport, physiology, signaling, and homeostasis work in these cells. And we're now in the process of generating the next generation, if you want to call it that, OnGuard 2, which will incorporate not just transport within the guard cells, but will incorporate photosynthesis, gas exchange, and basically allow us to go from the molecule in the guard cell all the way to the behavior of the whole plant in terms of photosynthesis, gas exchange, and water relations.
Excellent. Well, thank you so much for sharing this advice with us today, Mike. Is there any other piece of advice you want to leave our listeners with, or a last note of inspiration you want to end our conversation on today?
I think, well, for me, science has just been a delight, and to be quite honest, most often just plain fun. I get a huge buzz out of working through problems together with like-minded colleagues, and I've had the pleasure of working with quite a number of remarkably gifted people. I mentioned Chris Greffen before. I had a fantastic postdoc and colleague, Alex Grabov, many years ago, and Rucha Karnik, who's been working with me and has now started her own lab. And there are quite a number of others in between. I can't go through a long list. I suppose what I would say is that being a scientist and finding a vocation to build one's life around and get paid for it is quite a privilege. So as a scientist, I feel I also have a responsibility to pay society back as a whole. And of course, as an academic, what one does is to build knowledge and pass it on to the next generation. And I think most academics would probably share this sense of social responsibility as well. I think part of that responsibility is about being prepared to push boundaries of all kinds to build understanding. Of course, in this sense, traveling is quite important. We talked about that before. But I'm thinking not just sort of short trips to meetings and this sort of thing, but it's also important that scientists, especially young scientists, have an opportunity to work and train wherever their research takes them. Scientists and society would be much poorer without these opportunities. So I think the current populist and nationalistic trends that we're seeing right now across the West are deeply troubling. To my mind, they threaten to reverse much of the progress that we've made over the past 75 years. I think young scientists, and also older ones, myself included, always benefit from opportunities to work in different countries. And for my part, I've trained for longer periods of time in several countries, USA and the UK. I spent 1.5 years in Germany. I worked in Japan and China and also in Mexico. And my lab hosts PhD and postdocs from, well, over the past 30 years from about as many different countries. So I know that the lab also benefits tremendously from a group of people with very different backgrounds and often different cultures. And I think one of the things that scientists also can do is to help build bridges and understanding between cultures. And this is something that I would like to think that my lab and I think many others also do. So I think as a sort of a final note, scientists and science in an academic setting is a very powerful way of building understanding between people.
Certainly. Well, Mike, thank you so much for bringing up some of these really important and very timely points in your response here. And I think with all of these troubling happenings in the world today, we do have to, as scientists, have that responsibility in mind and try and work to make this world a better place, to keep being able to have these opportunities to not only support students, but to continue seeking these truths that we search for as scientists. So, wonderful message to leave our listeners with. Can you tell them how they can reach you to learn more about you and your work?
Well, you have my email address, michael.blatt@glasgow.ac.uk. And we also have a URL, which is www.psrg.org.uk.
Perfect. Well, listeners, definitely check out the website. Mike has some amazing research up there, and you can learn more about him and his lab and get in touch if you have any questions. And Mike, thank you so much for joining us on the program today and sharing a piece of your story.
A pleasure.
Well, it was a pleasure to speak with you as well. Listeners, thank you for joining us. We'll see you next time on another episode of People Behind the Science. Your voyage to explore the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to chatting with you next time on People Behind the Science.